Welcome everyone to today's CSAC Directors Seminar with David Relman on red lines involving research in the life sciences. My name's Drew Endy. I'll do a quick introduction and help moderate after David's remarks. Having courage to say no with wisdom is one of the most challenging things to do, especially in science. And we couldn't be luckier than to have David here to lead us through a consideration of that. We know David as the Thomas and Joan Merrigan Professor in Medicine, Professor of Microbiology and Immunology, Chief of Infectious Diseases at the Palo Alto VA, and Senior Fellow CSAC FSI. I know David better still through his time with the National Science Advisory Board for Biosecurity, the Committee on Science and Technology and Law, and other service roles in support of the public good. And when I reflect on those interactions, I first admit I'm lucky to know you. And second, wanted to offer 4 words, and just 4 words to help frame my confidence in David leading us through this challenging topic. In my experience, David is courageous, he's honest, he's tenacious in holding power to account. And in all those things, he's also kind. And so again, David, just really grateful and excited to learn from you today, and welcome everybody to the discussion that follows.
First, and most importantly, let me thank Drew for what are very generous and kind remarks, and also important for me because I had not until now thought of myself as Dr. No, but now I have further insight into possible roles in life. Well, that's certainly not my intention, is to play that role, but rather pose some questions and gain your wise counsel at the end of this, because I will add to these introductory remarks by saying, not only am I delighted to be here, but I come before you without certain answers, at least in my own mind, and much more so the certainty of important questions that haven't yet been adequately addressed. My outline here is to pitch the idea of the need for red lines in life sciences research, and to address the why now. But again, offering that I'm not fully convinced, but again, I do think this is the time to have a hard conversation about, if not, then why not? There are interesting lessons from the use of red lines in other domains, and for that, I do hope that you will either correct my misunderstandings of how they have been used elsewhere, and perhaps even better, offer perhaps more relevant analogies. And so I'll leave you with some conclusions and a proposal, and ask for your thoughts. I'll start by reminding you that it was only February of 1975 in Asilomar, California, down the coast here, that most people would time the beginning of the modern era of biology. It was the gathering of roughly 140 individuals, which included nearly all of those in the world who knew anything about manipulating DNA, i.e. the idea, the fantasy of altering life at its fundamental level. And even then, it was largely a dream. It was a concept. There were a few who had begun to piece together the tools that were needed, but that was 1975. And by simply 25 years later or so, so much had happened that it's in many ways mind-boggling. I'm not going to attempt to cover anything but a tiny little piece, an example of what happened over the first 25 years, and then subsequently, just to make some larger points about considerations here. But almost 25 years later, this paper appeared, and it was 10 years after the first paper that described a method for recreating a virus from simply a DNA sequence or an RNA sequence in the case of these viruses. The idea was to reassemble the genome, the DNA molecules necessary to boot up these self-replicating things, viruses, and how to do that in a laboratory. The first in the 1980s was the rabies virus, roughly speaking. And so it was 25 years later that influenza joined the increasing list of viruses for which there was a so-called reverse genetic system. And this led to the general notion that with a sequence, one can remake a virus in this case, and increasingly other things as well. Certainly with a sequence, begin to understand, begin to modify these other forms of life. Over the subsequent years, all of this has become more widely applicable and more easily used, illustrated by many examples, one of which is this paper published 2 years ago in response to the demand for the ability of many researchers to remake SARS-CoV-2 virus from chemical building blocks in order to understand it better, anticipate mutations that might confer resistance to drugs or vaccines, et cetera. And with these kinds of protocols, increasing numbers of people then became able to undertake this kind of work. You can perhaps make out at the bottom here, this protocol will enable researchers from different research backgrounds to master the use of the reverse genetic system and consequently accelerate COVID-19 research. And I just want to be very clear, nearly everybody who has thought about or gone down this path or made use of these sorts of techniques does so with the idea of trying to better our circumstances as a species. And there are countless examples. I'm just cherry picking one here with an early example of how, in the case of influenza, vaccines might be more rapidly produced in order to stay a little bit ahead of the annual evolution of circulating human-adapted influenza viruses and the need for new influenza vaccines every single fall, wherever that might be in the year, around the globe. And it's with that background that some important questions were asked when a series of naturally occurring outbreak events began actually in the mid-1990s, but then started to accelerate in the early 2000s. And those events took place in each of these countries, colored red, and were marked by the appearance of a brand new serotype of avian influenza. These are influenza viruses very well adapted to birds, not so well adapted to humans. The thing that was unusual and both interesting and concerning about these viruses was the degree to which they were well adapted for spread in bird populations, but more so the consequence of an accidental introduction of these viruses from birds to humans. And the result of those introductions was quite dire. Maybe can read, maybe can't, but in each of these cases, the odds of a fatal outcome were roughly 50%. So this is a virus that you could say is not well adapted to humans. No virus seeks to or is served well by a case fatality rate or an infection fatality rate of 50%. But that was what was observed in these relatively small numbers of cases. And yet, in each of these cases, there was no or very limited evidence of person-to-person transmission. And that was what then raised this important and problematic question: why hasn't this form of influenza virus, a virus that's eminently capable of rapid evolution, rapid adaptation, Why hasn't it become more transmissible among and between mammals? And, and because years had now gone on without any obvious evidence of that, is it even capable of this? And the answers were thought to be important because if the answers to the second were yes, and it simply hadn't had time in answer to the first, then it would behoove us, one thought, to try to anticipate what it would take, what it would look like, and what circumstance it might occur, and prepare. And so, in about mid 2000s, closer to 2008 or 9, the NIH, upper left, decided that it was within its mandate and purview and purpose to not only encourage the asking of those questions, but facilitate the answering of them. And they did so through a number of different mechanisms at their ready. One is to solicit investigator-initiated proposals, and the other is to issue contracts. Simply ask people to test or explore or do something. There are at least two examples that I'll highlight if none of this has yet rung a bell. The first was the awarding of a grant that was investigator-initiated by Yoshikawa Oka at the University of Wisconsin in Madison. Kawaoka, well-known influenza molecular biologist studying its pathogenesis, he decided that he was going to try to see if H5N1 could acquire greater transmissibility properties between mammals by taking one piece of the virus, the hemagglutinin gene. There are 8 genes. He decided to propose taking one and mutagenizing it, creating thousands of new sequence variants and putting all of those variants back into a parent virus, which in his case, he had deliberately crippled, so that the result, he thought, even if it were to be more transmissible, it might be less virulent than the starting virus. At the same time, the NIH decided to award a contract to an investigator named Ron Fouchier at Erasmus University in Rotterdam, Netherlands. And in this case, they asked Ron Fouchier, eminent molecular biologist, to see if he could create a more transmissible H5N1 using the best available mammalian transmission model in the laboratory that is known about, which is the ferret. And so Ron Fouchier decided that his approach would be to take the very viruses that were circulating in Southeast Asia with 50% case fatality and mutagenize the entire genome and see if he could create the actual thing, starting with the extant virus from nature with this high pathogenicity, and now see if it could acquire greater transmissibility, thinking we need to know what the end result actually might look like, if it's even possible. Will there be trade-offs, et cetera, perhaps transmissibility for virulence. And so he undertook the work. And at the time, none of this was reviewed other than by program officers within NIH. And so in 2012, when each of them produced a manuscript ready for publication, much of the rest of the world learned for the first time that this work had been undertaken. And the purpose was clear, the means and the net result and what to do about it were the pressing questions. And for those of us on a board that we had thought would have been asked ahead of time, what do you think about undertaking experiments of this sort, we were somewhat surprised to be reading about the results in manuscripts that Science and Nature had already said, we're ready to publish, we just need a go-ahead. And to make a very long story short, the paper was published, as you can see, and with it, the sequence that took Ron Fouchier 3 years to devise. But now, remember, have sequence produce virus. And the sequence is in the paper, and now the paper is all over the world. And so whether or not, and there was a great deal of controversy about this work, whether or not this actual virus that he created had all of the properties that we most fear, and whether or not what he learned from those experiments would materially change our planning for the possibility of an H5N1 pandemic, which was already being addressed by this point, is debatable. But one thing that is not debatable is the impact that this work had on public perceptions of the work of life scientists. This was the lead editorial in the New York Times the first Sunday after the announcement of these papers. And if you can't read it, it's an engineered doomsday, is what the New York Times declared. Scientists financed by the NIH have shown in a laboratory how that could happen. And this, of course, created a firestorm and a huge amount of rancor and discussion and hand wringing and hair pulling. It's all those sorts of things. And I won't go into all the detail, but suffice to say that this, to me, was the first moment when I thought, maybe we humans have actually arrived at a point, starting with 1975, when we actually could do something that might affect the fate of our species, or at the least, harm a fair number of people through a means that we might not be able to control. And this was the beginning, I would say, of an era, dates back now just a decade, in which increasingly frequent examples of work of this sort have been presented to us, again, all with the purpose of trying to provide better insight into what's happening in nature, what can we do about it, what do we need to understand better. An example is the Global Virome Project. Its purpose is to, up there at the top, capitalize on economies of scale in viral testing, systematically sampling mammals and birds to identify current unknown, potentially zoonotic viruses that they carry. Zoonotic meaning animal-borne, possibly transmittable to some other species like us. And they point out how many millions of potential viruses there are in nature and the imperative that we understand what's out there better. Now, I think we could have all kinds of interesting conversations about moral, scientific, practical rationales for understanding our world and what is there. But the question is, what do you do after you have a million new sequences, the kind of thing they are proposing? Are we smart enough to simply gaze at what we've observed and make predictions about which of these is going to be the next 1918 flu? And unfortunately, the answer to that is no, not really. We are not yet able to go from sequence to full predictive powers of that sort. And so scientists, having realized this, then say, okay, and so said the authors of the Global Virome Project from around the world, ranging from UC Davis all the way to Wuhan, China. They said, okay, we're going to have a plan for down selecting the sequences of greatest interest. What can we do to clarify the functional properties of these sequences? So here's an example. One of the families of important viruses that we all recognized back in the 2000 teens were the coronaviruses. They'd already made their presence known through pandemics previous. And the Global Virome Project and all the other scientists out there that were looking at diversity in nature were already starting to find lots and lots of interesting coronavirus sequences. Some of them interestingly close to known pandemic causing viruses, others total toss-up. We just don't know what this thing might be capable of, if anything, for a human or for any other host other than the one from which we found them, in this case, bats. And One thing was known, though, about coronaviruses, and you can thank this insight for the fact that hopefully all of you have now received um, a COVID vaccine. It was already known in the 20-teens that the key part of any coronavirus is its spike gene and the spike protein. That's the piece of the virus that mediates encounters with host cells. And so they said, hey, we've got a bunch of interesting spike protein sequences. How are we going to tell which of them might be most interesting or important or biologically significant? And this was the approach. They said in this paper, we're going to take the spike protein gene, that gene alone, and we're going to put it into another virus whose properties we already know a little bit about. And we're going to see if this new chimera is either more able or less able to infect a human cell, more or less able to grow to high titers, high numbers, more or less able to spread. And a well-recognized approach. The point, though, here is that the starting virus, the template into which they decided to plug all of these new spike protein genes, was a virus called WIV1 that had also been discovered in China in much the same locations as these new bat coronaviruses. This too was a bat coronavirus, WIV1. It had already been studied, but what had been learned by Ralph Baric and others, I would say the world preeminent coronavirologist, was this virus, shockingly, is already well adapted to human cells, straight out of nature. They didn't do anything except synthesize it from the sequence as is and ask whether it could infect primary human respiratory epithelial cells, which is a pretty good model in vitro in the laboratory. And lo and behold, they found it was poised for human emergence. And in the discussion of this paper, Ralph says, whoa, this is a virus that we need to pay attention to. Let's be careful when we work with viruses like this. So off the record, I will tell you that Ralph was quite humbled and somewhat surprised when he saw this paper, because what in essence they were doing was shuffling a bunch of spike protein genes with unknown consequence into a virus that was already teetering on the brink of being able to be off to the races with humans. A bit like Russian roulette, you could say. Now, what made it a little more concerning and politically charged was, at the time, in China and elsewhere in the world, until you could know that a resulting virus would have dangerous properties, you were okay, said authorities, in operating at a relatively low biosafety level. That's BL2, and that's the level at which they did this work. And then the politically charged part is, this work was partially funded by the NIH. And so we now have, a whole body of work growing in numbers and power and potential significance for the good around the world that risks possible outbreaks. This is actually part one of a two-part series that came out in the Washington Post on Monday, was this one, and on Wednesday, yesterday, was this one, which provides the counterpart story. We're really not so swift at biosafety, as it turns out. Another investigative reporter by the name of Allison Young had already shown over the last 10 years through Freedom of Information Act requests to biosafety panels around the United States, including our own, that there are many, many transgressions, accidents that are not reported to the public, and in some cases not even reported to the NIH in a timely fashion. This one that I'm cherry picking happens to be Yoshikawa Oka's lab, the one that was tinkering with H5N1 years ago, is now working with a more modern version of an engineered H5N1. And one of their investigators, this is December of 2019, just as COVID is blowing up, they realized that at the end of the day, they'd been working with this virus in a laboratory, and the hose that attaches to the face mask that's supposed to be providing purified air, the hose was unplugged. It was just dangling in the wind. And so potential exposure. But NIH doesn't hear for days. And during the subsequent negotiations in private, there was a joint decision that this probably wasn't a true exposure and that not much needed to be done. And certainly we don't need to worry the public about things that we think we're better able to judge, said they. So all of this has taken place in the background of ongoing discussions about what do we do about work of this sort. And in the wake of the H5N1 kerfuffle in 2012, there were a lot of conversations about how to manage the possible risk from these experiments. And the net result was that HHS, Health and Human Services, through NIH, and the White House, OSTP, Office of Science and Technology Policy, decided that it was time to have a little pause, a little timeout. And so that was announced in October of 2014. And the announcement said, the United States government will institute a pause on funding for any new studies that involve certain so-called gain-of-function experiments involving influenza, SARS, and MERS. These were the only respiratory viruses that they thought deserved this kind of special attention. Gain of function here referring to experiments whose net result would be to add new capabilities to something like a virus, but especially transmissibility and or virulence. So time to pause. And during this time of pause, we're going to have a lot more deliberate discussion about how to understand and manage these risks. 3 years go by. This is now December 2017. And the NIH, in collaboration with the rest of the US government, decides, we think we have a plan. We're going to call it a framework for guiding funding decisions about proposed research involving enhanced potential pandemic pathogens, or EPPP. And this plan, this framework, was announced at the time that they said, okay, we think we can now resume funding, but with the guidance from this framework. And I'll just show you, again, probably can't read. There are a couple features to the framework. First of all, they define very narrowly what it is that ought to prompt a more closer look. It's going to be work on these particular subset of viruses that have the potential to be enhanced in such a way as to create a possible pandemic risk. And the process is going to entail a review by local institutions in some cases, but NIH and HHS in most cases. And they're going to look at risks and benefits and make some decisions about what to recommend. Again, the scope is narrow. The range of possible outcomes includes the possibility of not funding research. But what they don't say is what they would then do about a proposal that might be viewed as excessively risky. In most cases, though, the options available are going to be fund with modification, fund with modified communications plan, have further discussions and fund, et cetera. And the net result of this entire process now, the framework that was put in place in 2017, taking us up to today, as of today on the NIH website, only 3 projects involving this kind of work have even been reviewed. Only 3 were felt to be deserving of this kind of review. They did not include, for example, the Russian roulette paper that I showed you. And there are many other examples that were not there. What they don't tell us actually is what 3 these are, who looked at them, what the nature of the deliberations were. It's all opaque. But the outcome in all 3 cases was, you're good to go. except in one where they said, let's just focus on one part of your experiment and not the other. So that's basically, with the following exception, where we are right now. Last year, the NIH, NSABB, this Biosecurity Advisory Board, the White House decided maybe this framework doesn't satisfy all of our needs. There'd been a lot of criticism about the fact that only 3 projects had been reviewed. There was criticism in the form of a paper that Megan Palmer and I and colleagues wrote to say, this is excessively narrow. The range of options you're considering is very narrow, and the process is impossible to assess because we don't even know what you're doing. And so a proposal's been made as of this past January to expand the scope of that framework. And that's basically what this says. We're going to consider a much wider range of work, but what it doesn't say is, here's a list of new possible actions and intended purposes. And it doesn't say, for example, we're now going to be mindful of the fact that some work may be safe where it originally done, but not safe where it might be revisited by someone who now has a sequence. So it leaves us essentially back where I think I may have shown this slide here in this room about 10 years ago, which comes out of Jurassic Park. But your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should. So what are we going to do? There are a bunch of options here. Some of these are sort of minimally risky to the enterprise of science, and for very good reason. As you've probably seen from the public discourse, there are now a number of actions that would include a complete clampdown or shutdown of much of this work from the start. We're just not going to do any of it at all. And again, for reasons that the public has every reason to propose or endorse. Doesn't sound like we scientists are acting in a very responsible fashion, let's say. But we could continue down the path that the NSABB has begun or recently revisited, which is further refine the scope of work which will now be reviewed using perhaps better forms, more quantitative forms, if that's possible, of risk-benefit analysis. Much to say there. But review that would still have to, at the end of the day, result in actions that, to me, have not yet been well articulated. And still, I think we have an unanswered question. At the end of the day, is everything actually okay, as long as we're just mindful and careful in how we talk about it and explain it, and the number of biosafety precautions are undertaken, et cetera? Is it all basically going to be okay or not? Because it would appear that there is nothing yet that finally warrants not just undue concern, but action. So that leads finally to the issue at hand, red lines. And this is where again, like, you turn on your critical hat and think about your understanding of how red lines have been used, to what avail, et cetera. Because this is where I start down a path that to me seems potentially promising, but fraught, clearly fraught. First of all, we're not even clear about what does a red line mean. The term has been used in all kinds of different fashions, and it's been used or substituted with other terms that are meant at the time to reflect similar kinds of concerns, things like lines in the sand, or thin red lines, et cetera. And you start to kind of devolve into a much more murky set of terms and purposes. And so that's one issue that clearly has to be addressed here. But I pulled a few examples out of history that I thought were just useful, informative, et cetera. This is one that is often cited as the first clear, explicit use of a line to affect some kind of decision or action. it was the occasion of the armies of the king of the Syrian Empire at the time, Antiochus, who had now taken his army into Egypt and was about to lay siege to Alexandria, prompting concern from the Roman Senate, and for fear this was going to destabilize the region. And so the Roman Senate sent an ambassador to meet with Antiochus, and the ambassador carried some tablets with orders, basically, from the Roman Senate. And the order said, leave Egypt, or else. Not articulated, just it would be a good idea for you to turn around and take your army out of here. And apparently, the tablets were handed to Antiochus, and he read them and he said, okay, if I get back to you. And the ambassador, Pompilius, said, actually, no. And then drew this line around him in the sand and said, you're not stepping outside this line until you give me an answer. And it better be the right one. So the king apparently said, fine, I understand your perspective, we will leave. And so he did. There's another interesting example that it almost turns this whole idea of a line in the sand around in a different fashion. So this is the story of Sita and Rama in what's called Ramayana. I'm, again, not a scholar here, but this is one of the most famous Sanskrit writings in Hindu culture. Again, around the same time, about 200 BC. Here, the circumstance is Sita, who is a Hindu goddess, and goddess of wisdom and groundedness and understanding of the earth, etc. She has now lost her brother, who was beckoned out into the forest because of sounds of some animal in distress, only to have it turned out that the sounds were made by dastardly evil forces. And he's now disappeared, and she's very upset. She calls upon, sorry, it's her husband that has gone off. She now turns to her brother, that's not him, and says, could you please go out there and find Rama? And he goes, okay, I'm going to leave, but that means you're alone. And so what I'm going to do is draw a line around your home, and the line is meant to protect it from these evil forces outside. And now what apparently was to happen is anyone other than Sita and Rama, he's missing, and the brother Lakshmana, anyone other than those three will burn to death if they cross the line. But here, the difference is the purpose is to prevent badness from coming in to a good part of a good domain. And it does allow people, the chosen few, to leave, but at their own peril. So think about that as another way of formulating the value of a red line or a line in the sand, meant to both protect and avoid outside harm. And then the third, which always gets called upon here, will be well known to all of you. It goes back to, again, 2012. Seems like all the important turning points happened in 2012 when Barack Obama said this: we've been very clear to Assad in Syria, there is a red line that would involve seeing a bunch of chemical weapons moving about or being used. And as you all well know, during the course of the subsequent year or so, certainly by spring of 2013, there was growing evidence of Syrian use of chemical weapons, until finally, on almost a year to the day, there was clear documentation in Ghouta, this suburb of Damascus, of a terrible atrocity, 1,000 people dead, of what almost certainly had to be chemical weapons use. And then there were a lot of machinations, a lot of struggle within the White House to now decide what do we do. And there are really interesting accounts of how this all played out. Ben Rhodes, who was the Deputy National Security Advisor at the time, was actually tasked as being the proponent for an action to take against Syria. And he built a case and then presented it to the rest of the national security advisors, et cetera. But at the end of the day, as you know, the decision was that we were not going to take at least military action. And the reasons, I think, are obviously complicated, but it clearly involved political calculus, because suddenly what had been said to be a clear, bright red line was now something that was fraught with politics and political consequences, which perhaps, it seems, had not been fully assessed or understood, one or the other, at the time the original statements were made. And so Obama ends up essentially backtracking and saying, I didn't set a red line, the world did. This is a norm that represents the thinking and will of the world's population. And so now he's essentially saying, I'm no longer the actor, or else I no longer have the authorities, for various reasons, to undertake an action or a response. There are other forces outside of my control. And as you know, the outcome was to defer to the Russians, who were more than happy to come in and try to manage the situation and seize sort of the lead. And so all of this starts to raise questions about, how do red lines work? What are the problems with red lines? It would seem that in recent times, in the political sector anyway, the purpose is often deterrence, or what some people would call coercive diplomacy. But it's imperative that there be clear objectives, clear circumstance under which the line will be drawn and understood, and clarity of response. In other words, the response to the transgression of the line must be understood ahead of time and measurable to make it real. And there must be commitment both to the threat that's implied, but also, as argued by a number of people, at the same time, the need for commitment to promises. And so there's this issue of credibility, which would include capacity to act, authority to act, the will, and the interests. And I started reading about how do people sort of assess the value of these things. This paper raises what I thought were some valuable little superficial lessons from recent times. Dan Altman and Kathleen Powers, they actually conclude, up here on the left is shown, red lines and threats more generally fail more often than they succeed. Why? Well, there are a bunch of potential reasons and problems. There's the risk, if not fully sort of executed as presented, of damaging one's reputation or credibility. Others would say, in reality, that may not be such a big deal, especially if it's understood explicitly why you weren't going to follow through on this one, as you suggested you might. But it has to do as well with the problem of asymmetric interests. If I care more and I'm drawing the line than someone else does, then someone else knows that I'm likely to follow through. But if they care more than it's known I care, then it's quite possible that no one's going to really believe me and perhaps even understand if I don't respond. And that could be the case today with Taiwan, for example, or maybe with Obama and the chemical weapons. Maybe, in fact, despite what I believe was true moral shock and outrage, and that of all of us, it wasn't important enough in the interests, as he saw it, of the United States, certainly wasn't in the interests of Congress, to follow through. There are clearly information hazards. People would have argued and shown in cases that if you're very clear about the thing that you most fear and you don't want someone to do, you may well prompt people to actually take a closer look and maybe even do the very thing that you wish they would not. And there's a lot of study here in the psychology of threats. And you can go to shelling and game theory. You can go to all kinds of examples where it's clear that, especially with irrational actors, we can think of a few today, and especially when the threat is adamant, public, and sort of extreme almost, that this is what may happen. You provoke your target. And then there's also the concern that if you draw a line here and enforce it, your adversary, your target, may now wonder, what's next? Are they now, especially if you haven't yet and you're considering conceding, if I concede, am I simply going to encourage that new lines be drawn? And I'm asked ever more and ever again to do things that I hadn't even thought about not doing until my antagonist decides to assert them. So it might raise the question, well, okay, if not a red line, are there any good alternatives? People who have looked at this term red line often end up concluding it really wasn't a red line. It was an ambiguous threat that was actually undertaken deliberately, strategically, in order to maintain a couple of advantages. One being, if I'm not really clear what it is that I most fear or don't want you to do, the actor can decide when and where I wish as to whether I will enforce it, but I maintain some uncertainty in my adversary, and they may hesitate because they're unclear. And it could work, again, the other way too. Some people have said maybe this was a good strategy for US and Taiwan, because if Taiwan isn't sure that we're going to enforce a red line upon a Chinese invasion, maybe they won't be so strident in asserting, their, fierce independence and provoke China as much. So maybe that kind of thing works under certain circumstances, but I think now when you look at some of those examples, perhaps it works only for a certain period of time under certain circumstances and not many others. So what are some other alternatives? Well, what about just general norms, principles, ethical principles? Thou shalt not do harm, without articulating exactly what happens if you do harm. But there's such a strong moral urging that perhaps the consequence is disrespect, is shame, the things that some people care about, perhaps not others. And finally, I suppose, especially in science, you hear this a lot, and I kind of get this. Why not just full speed ahead? We're going to emphasize the ways in which the science will defend us better, create good defenses, and simply hope for the best. Try to stay ahead of the risk, the threat. And so when you think about this life sciences situation, I'm sort of stuck with some issues here. I think, as I've already suggested, that we don't have good clarity right now on what, if anything, really matters enough to do something unusual with consequence. I don't think that strategic ambiguity necessarily is going to, save us in the end. On the one hand, it may allow lots of good scientists to continue doing innovative work without having to worry, because it's not clear that they're near a line or even crossing one. But on the other hand, I can't see where we don't just go careening off a road in doing so, without even realizing it until the paper's written. And the other problem here is, even if the work will serve our own defense, it's always the case that defenses take much longer to substantiate and implement than the misuse of the threat or the risk that you've suddenly now just made evident to everyone. There's a disconnect in time. And it gets to something that again, in reading about this, I got interested in exploring a little bit more this idea of a so-called cultural lag. So William Ogburn published this really interesting book, Social Change. This is 1923. And he was a professor of sociology at Columbia. And he coined this term, cultural lag. And what he meant was, okay, we now have, he says, it's the 1920s, the world is being upended by technologies and advances. And we humans evolve slowly, both genetically and culturally. So he says, this rapidity of change in modern times raises the very important question of social adjustment. The thesis is that various parts of modern culture are not changing at the same rate. He gives, for example, industry versus education. Industry is galloping ahead, education is lagging behind. Where one part of culture changes first through some discovery or innovation and occasions changes in some part of culture dependent on it, there frequently is a delay in the changes occasioned in the dependent part. In other words, we have this period of maladjustment and we ought to be thinking about how do we shorten it. And that, to me, I think is part of the challenge here, because you have science that poses something today that clearly has promise and peril, but the promise is not going to be realized for quite some time. The peril, I think, we're stuck with much sooner. And so I'm going to propose that we, if not give, if not adopt immediately some semblance of a proposed red line, that we give much more serious consideration of it or them. And I'll pitch one right here. I will say that one shall not deliberately create an agent or easily anticipate the creation of this agent not found in nature that's both highly transmissible and highly pathogenic. So it disseminates easily, it causes lots of harm. And you can just genericize and think about things that have both of those properties that don't exist in nature already. And the consequence has to be significant enough that we're actually going to do something special here. And the consequence here is substantial harm to be defined to all the things we care about, including our environment, but not to mention humans, animals, plants, etc. Well, this is a proposal. I'm going to argue that transmissibility is the key feature here, because something that's highly transmissible, now you've seen it, SARS-CoV-2, goes so fast, you can't get defenses out in front of it quickly enough. We don't even accept them when they're sitting on our shelf. Plus, you have the unequal receipt of risks and benefits around the world. Plenty of the world is pretty upset if you say, hey, maybe I'm going to make something that has potential downsides, but don't worry, we're going to have defenses available. Because they're going to say, in what century do you think I'm going to actually see them in my part of the world? So one advantage, though, of a red line is maybe it just buys time. It allows us to shorten the cultural gap. It allows us to further strengthen norms, which might already be there, and discourage harmful behavior. But mindful of the fact that you don't want to be so specific that you've actually disclosed exactly what you don't want someone to do. And now, what are the objectives of it? Well, you signal that there are actually lines, which is something our government and no other government has so far been willing to say. And sometimes when you argue this with policymakers, they say, fine, but give me an example of something that might be that. And I said this to somebody once. I said, what if you made HIV aerosol transmitted? And they said, well, that's not going to work. You can't make HIV transmittable. I said, well, that's something we could argue about. Do you want someone to try really hard with today's technologies? And they said, no, I suppose not. But who would do that anyway? Because all scientists are good people. All that I've ever known, said this person, are good people. So maybe all we do is reduce the number of times that people cross over a line, because at least they know it's there. But there has to be some consequence. And there have to be alternatives available to them that are rewarded. This is the the commitment to promise. And of course, it strengthens and makes visible our responsibilities to society. The consequences part, still a work in progress in my mind. There are going to be penalties and there are going to be promises. The penalties may be things that scientists care about. If the scientist is your potential malactor, funding, other resources, publication ban, shame. Maybe you're going to have to pay. You're going to have to pay for the consequent misuse. Could be liabilities, maybe even criminal penalties. But there have to be promises. Otherwise, this just doesn't work. And the promises are going to be, if you are mindful of the guardrails, of the lines, then you're prioritized for resource allocation. And there are going to be things we're going to do to make sure the public actually respects what you're doing for them in ways that we hope will work in your benefit. And I don't think this is going to work alone, but I'm just going to say, if you were to believe, or want to try the first three things or the first three features, I would say there still have to be some complementary strategies, other forms of speed bumps in the form of a periodic moratorium, but also guidance about how to stay down the sort of the center of the highway that we do want people to go racing down in science, using streetlights. What are good things that we would love to see you do, or good approaches to these really, difficult questions? And the governance part, still completely unclear. I'd love to think that the WHO is ready to take something like this on, but it is not. Whole nother discussion. But if not, then who? And does it need to be sort of balkanized necessarily and made local somehow and coordinated? I'm not sure. So here are my takeaways. Rate of innovation is amazing right now, with both benefits and risks at increasing pace. I would argue that our existing approaches alone are not adequate. We need red lines. It's my contention. But they must be carefully calibrated in some of the ways I just alluded to, but are going to have to, they deserve a lot more careful thought. And we're not going to rely on them alone. Say I. Okay, I'm going to stop there. I'd love to hear your thoughts, your criticisms, your comments, etc. Thank you for your attention.
Thank you, David. We have plenty of time for questions and discussion. If I'd asked for the folks who are on the Zoom, their benefit, if you could quickly introduce yourself and use the mic, and we'll just go around, and I'll loop in some of the questions from the chat as well. If you're on the Zoom and would like to pose a question, please use the Q&A function and we'll represent that to David in the room.
Thanks so much, David. Wonderful presentation. And, I loved all the historical analogies and things. One thing, though, that just, playing devil's advocate, that, I've had the pleasure a couple of weeks ago, coming to an amazing talk by actually one of, former CSAC fellows, who, was on the front lines with trying to get North Korea to, basically get rid of their nuclear weapons. And, it's not worked. And despite, the fact that, there's been very heavy sanctions, on that country. And, I just may be a little discouraged that, particularly in biology research, that, we all know how easy it is to order oligos and to do site-directed mutagenesis, even old technology like that, and that what I just don't really see that we've been able successful in these other more visible areas, to really influence people doing bad things.
Thank you, Dean. It's a great question, and you touched on all the sort of many of the key issues at hand. And I agree with you. SIG did a masterful job of making the case for some other approach, maybe not even this one. But I would say that what I took from SIG's talk and that whole history was that red lines were never properly undertaken. There was no possible way through the means by which they were implied or considered that they were going to be successful. And there are lots of lots of reasons that you could point to. One is that, first of all, the line kept changing and was not clearly articulated. But among the many other reasons, I think was a point that Sig made really well, which is that there never was a commitment to a promise. It was never going to be clear what there was to be gained by trying to adhere to this line, that there would be a new and better world available. It was only the absence of pain, I think, was his message of what was perceived. Now, maybe there were intentions to follow through with promises, but I don't think that, certainly after the first few visits and first few years, that was likely to have been clear. And I think that's again, a more general kind of problem that red lines have had. They appear to have failed when in fact they weren't done in a carefully considered, they weren't carefully calibrated, as they say.
Tom Finger from A Park. Thank you, David. You've clearly put a lot of thought into this, and I think kind of moved the subject forward. What the presentation suggests to me is a nesting or an onion-like set of layers or things inside one another, that beginning with your sample prescription on transmissibility and lethality as an ethical point that, responsible scientists don't do this. A next layer out in things like funding organizations will not fund research that goes into those areas. Another layer begins to get into what are the consequences. We need all, in quotes, of the principal outlets for scientific publication and membership in societies and so forth that would adopt rules that they're prepared to enforce if somebody does this. The tricky part of use of red lines, I don't even like the terminology coming out of the world that I do, is that there will be people who, once the line is decided, that's an attraction. There must be a reason they don't want me to cross that. There must be rewards if I am able to cross that. And that kind of thinking, it's hard for me to imagine what a sanction demonstrated or stated in advance would deter. So I think you're on the way to developing a set of guidelines that will reduce the risk from responsible scientists doing responsible work, with feedback loops in there and reporting requirements if this unforeseen development occurs during the experiment that feeds back in, and that doesn't devote a lot of attention to the outliers who, end of the day, would probably do it anyway. But I think that's what's inherent in the presentation that you set out.
Thanks, Tom. I appreciate your comment for a couple of reasons. One is I think you've framed the issue as basically, to me, the admission that we are flawed beings living in an imperfect world, and we're never going to escape that. So I hope I didn't suggest that I think we do all these things and we're good. And I know you don't think I said that either, but I think that, having said that, if we simply reduced the numbers of people who were willing to go down or go, veer towards the edge of the road and stare over the precipice, or even, belay down just to see what's down there, so much the better. And there will for sure always be people who are hell-bent on doing exactly what you wish not. But I would say that as long as you don't present information hazards, and as long as you have promises, you can counterbalance any, incremental additive harm that may come from this effort. Other than perhaps the rapid, decision that these people are foolish for thinking about this, and I have even less respect for their wits because they even thought this could do some good. We'd have to measure. We'd have to show what it is that didn't happen as well as what did. But at the end of the day, all of us have choices to make. When we get up in the morning, and the reasons why we pick one thing to do today and not a bunch of other things is complicated. And all I'm saying is let's just complicate it further. Let's tip it. Let's bias it a little bit. That's all.
Tom Burton from a CSAC affiliate. Thanks, David, for your talk. I couldn't help seeing the strong resonance between the story you're telling in life sciences and synthetic biology and the story that's also salient in the news right now about advances in generative AIs. And everybody who will be aware that last week there was a proposal, a letter signed by many people saying, hey, let's have a timeout on these developments of these large language models. Let's stop at GPT-4, not go to GPT-5 until we have a chance to think about it for a while. So I think that in life sciences, you're ahead of where people are in the AI world because you've already tried this timeout. You've already tried a framework. You already have a hand on the purse. The government, the AI work is not being funded primarily by government, while life sciences is. So I think you have some advantages here, and I think you're ahead and a pioneer. So we're looking carefully at what you do.
Tom, you're way too kind because I think maybe the response is we're really doomed with AI, but I don't think that. I think we're behind other disciplines. I thought maybe you'd pull out, world of nuclear physics, where they have the advantage of every new generation of scientists already having been born into a world in which it was pretty much understood. There are things that we agree we're not going to do. We're not going to proliferate the design for a bomb. Now, people would love to, but generally, I would argue, I have no evidence of this, that it has happened far less than it might have if, yesterday we discovered that you could do such a thing, and now everyone's really curious about it and needs to learn on their own and decide on their own. In physics, I think there's already some good history there. And I maybe with time, maybe another positive statement is with time, as we life scientists learn to kind of accommodate and adjust to this reality, we too give rise to new generations of people who are already having it baked in somehow.
Yeah, Scott Sagan, co-director here at CSAC and professor of political science. I thought this was a real model of what we try to do at FSI, was bring people from different areas together, talk about ideas and borrow ideas and learn from each other. So I was thrilled when you started and cited Dan Altman, who wrote his Red Lines PhD thesis here. It was MIT PhD, but wrote it as a pre-doc here. And I just, in that spirit, I wanted to give two comments that I think could be helpful in the next iteration of this. One is that in law, there is a difference in what people will call a rule versus a standard. A rule is you can't drive above 65 miles an hour. A standard is that you can't drive dangerously or irresponsibly, right? And a rule is a red line. It's a bright line rule, and it has the danger of under inclusivity or over inclusivity. You say you have to be 18 to vote. But we all know people who are 16 who are smart enough and knowledgeable to vote responsibly. And we all know people who are 35 who probably shouldn't be voting for lots of reasons, right? But we draw the line because it's easy to execute, and it means that people can't manipulate it quite as readily, even though we understand that may not be effective. And when you talked about your red line rule, it wasn't a red line at all. There's a bunch of standards that left enormous room for interpretation. Highly pathogenic, can't be easily stopped. Those are words that you use when you want to have flexibility. So I think in terms of thinking about standards and rules, that would be an area in which talking to the lawyers who deal with that conceptually would be very useful for the next step. And the other is that, for lack of a better term, half of the problem of a red line principle is the clarity of the act that you're trying to stop. That is, what is acceptable, what is not. The other is the clarity of the response, or the certainty of the response. So it's not a red line, it's a red response, or it's the commitment to response. And here there's a literature in political science about audience costs and commitment traps. That an audience cost, that when you make a commitment that if you cross that red line, I will do X, that it does not just communicate resolve, it adds resolve, because it creates a condition in which if you don't do that, you will either suffer domestic consequences because you'll be considered weak and not following through on your threats, or you will increase the likelihood of future things because internationally you'll lose your credibility. And so there's a whole debate about under what conditions do people actually suffer domestic audience costs and not. So here I think in your area, it would be very interesting to study what are the punishments. You have a whole list of kinds of things. Well, what do we know about what's worked in that area and not? When have scientists actually been constrained by their beliefs that there'd be a punishment in terms of loss of prestige or loss of funding versus when not? So I think this is a really rich area, and I'm really excited about it. And I want to introduce you to some of the other political scientist people here who are working in this kind of area, because I think it's super promising.
Scott, thank you. This is exactly why I was really eager to do this, with all humility, because this is exactly the kind of, deeper thinking about this that I would love to explore. I did note just a couple quick responses on the standard versus rule. It turns out that the Indian parliament has adopted the very same term, that Hindu Sanskrit fable, that line, they use that name as a standard actually for operations in their parliament. For example, they say the judiciary and the legislature should not, pass a certain line for fear of the following consequences, and they apparently stick to that, and they treat it as a legal standard, apparently. So I'm probably botching or not fully reflecting how these are used in true legal terms, but I think that's a really important distinction. I'm going to look at that more on the second point about I'm trying to remember now. Yeah. So this is where your student Dan Altman has a commentary in that foreign affairs piece about what he says is the overplaying of the credibility damage argument against red lines. He says, you look at all these examples, and although you would have, on the face of it, said, what a terrible thing to do, Barack Obama, you've lost further credibility. The reality, says he, is that the context in which you seemingly backtracked or failed to act was actually explicitly understood and made clear. And there was some harm, but not nearly as bad as people have argued. And instead, there are other things that you ought to be careful about and be mindful of, like the promises, for example. Yeah, it's a lot more.
Steve Luby from School of Medicine and CSAC. Thanks a lot, David. I also salute your willingness to address this area, which really just seems to be not talked about nearly as much as it should. Just a couple thoughts. I question whether the whole terminology red line and establishing red lines and trying to develop the whole system is actually the best way forward. And maybe this just reflects a little bit of my endorsement of Tom's suggestion and being a little bit more of an incrementalist. So how about instead of big talks about red lines and comparisons with North Korea, we instead put up the Relman proposal of saying, let's not advance novel viruses outside of nature that are increasingly transmissible and virulent. And the NIH will not fund this. Because that's a place that we might be able to start and get immediately achievable. Might we also say, let's broaden our review of pathogens of concern beyond the narrow concerns of biosecurity in the lab, to step back and say, what's the broader social risk associated with this? And can we have that weigh into whether or not we go forward? So it just seems to me that if we could take some steps that are probably easier to move forward and even get some comfort with that, then we might be able to say, well, are there ways that we could further strengthen and add more sanctions? But it just seems to me that trying to do everything all at once is very difficult in this moment.
Thank you, Steve, for the voice of reason and reality. When I was a freshman in college, I was I know we have some MIT grads here, right? At least one. I was course 17, so I was political science major. And we had an intersession opportunity to go to Washington, and I was assigned to work with a guy named Robert Drinan, who was a Massachusetts Democrat, very prominent for his role in taking down the House Un-American Activities Committee from the inside, et cetera, et cetera, et cetera. And I had lunch with Drinan sort of every other day. And when I was there, one of the things he did was he sued the US government for bombing Cambodia. He was out there. And I said to him once, I said, you know, I'm told that you never got a bill out of committee, let alone onto the floor for a vote. You know, how do you assess your impact here? And he said, my purpose is to simply shift this huge thing about 2 millimeters this direction, and I'll be happy. Because that was what he thought his role was. So I'm almost feeling like maybe I don't want to be just, you know, carelessly provocative, because I do really believe that this is needed. But to your point, I think you're right. At the end of the day, we have to try something. And I already know that half of my colleagues across campus think this is foolishness times a million. So gonna have to make some accommodations for sure.
I'm tracking 2 additional in the room. There's 2. I'll read in quickly once we're through. We've got about 10 minutes left. And I'm going to give somebody else, the moderator, stick, because I want to offer something, but I don't want to have 2 responsibilities. So here, I'll read in the Zoom, and then
I'll try to be quicker in my response.
we're good.
Yeah.
David Holloway here at CSAC. David, thanks very much for this talk. Extraordinarily interesting. I mean, I'm not in the life sciences at all, but it does raise a lot of parallels, I think, with, let's say, with the nuclear area, Right. So, but really, there are two things I wanted to ask you about. So, one is you're really talking to or about a set of institutions, so funding agencies, editorial boards. I don't know now with open access, how small or big communities they are, to begin to perhaps review more carefully or agree on different criteria for assessing publications. I mean, in line with what you propose. I mean, it did happen in the nuclear field very early in about 1940, 41. Journal said we won't publish any more on nuclear fission, essentially. It didn't stop the bomb being developed. But so that was one thing, whether some of the presentation could be angled more towards the group of institutions that have some authority and some role in this area, funding agencies, editorial boards, and also probably to start in the US, but this is also an international problem because that you know, for reasons already raised. The second thing that I wanted to talk about, and if you think about nuclear proliferation, there is a critique which ties in with what Sig was saying in his talk, and in his book, he emphasizes the great importance of diplomacy not being well practiced. It's more, we lay down what the lines are, and if you don't agree to them, then we'll punish you in some way. But it seems to me in this area, that also would be extremely important, because the punishment will be not to governments, it'll be to individuals or particular labs or something. And there are enough people with money around the world who, could fund people to do things if government funding was cut off. So I think that's an element of, I mean, it's a question I have really, and what you say about your colleagues' attitude to what you're proposing seems that's a big challenge to get a buy-in from people on this.
Yeah, I was a little glib in that last comment of mine, but no, you raised some really good points. I'll just say briefly, the institution issue is being addressed by a number of groups right now. For example, the Nuclear Threat Initiative has a big program to address the publishers. But frankly, with self-publication becoming the norm, we're no longer dependent upon or under the control of these external entities that once, held a tight hand over all of this. Although I'll say that Ron Fouchier said at the time, or soon after the ruckus arose, he said, I regret very much pursuing these experiments or talking about them the way I did. I simply did so because I frankly was looking for some attention from the high visibility journals. Wanted a headline. It's good for funding. If science had said to him, sorry, this is just irresponsible, and we're going to tell all of our peers not to publish it. Well, at that time, 2012, that might have actually have worked. But today, I think you're right. And so I'm thinking further back upstream, at the point of the inception of the idea. It's Monday morning. I've got a pressing problem. There are 5 ways I can address it in my lab. One of them has undue risk. The other 4 are okay, maybe not quite as effective. Maybe I should pick one of the other 4. And then, the last part on what are the actions that matter. I think, Scott's point about, or somebody said, let's try to understand better what people would respond to and how you would do it in a negotiated fashion to get to your point about diplomacy. I agree with that as well.
Hi, I'm Miriam Barnum. I'm a postdoctoral fellow here at CSAC. Thanks for this talk and for making it very engaging and accessible to those of us who don't have a life sciences background. So thinking about the sort of political science literature on red lines that you're drawing on, I think one element that's very emphasized by that literature is the actor that's doing the enforcement and the credibility of that actor, and things like that. And often when you're talking about these red lines, you're sort of talking about we should have red lines, we should do X, we should do Y. And so it seems like it implied in some places that the actor there is either the US government, funders, or sort of scientific professional organizations and journals. But when we get to the issue of sort of trying to limit these risks in, for instance, other countries where the government might not be as interested in trying to support these sort of red lines or impose them either for malicious reasons or simply because they want to develop their industry quickly and they don't care about guardrails. Sort of what should be the actor doing the enforcement in that case? Should there be international organizations that do this? Should individual states try to do this, as with like the Obama Syria case?
Yeah, great points as well. And David raised the issue of internationalizing, viewing this as a global issue, problem. I agree. There probably are existing institutions that are international in nature that could have some modestly useful role. For example, scientists care about the respect of their peers and their standing in the scientific community. One of the more respect-generating institutions would be academies of science, professional organizations that matter to people, maybe not to some generations now, but they have. And there's an inter-academy panel of academies of science around the world. So for example, that kind of internationalizing of the kinds of institutions that we think matter to most people might be helpful. Wouldn't solve everything for sure. But increasingly, this science is a global enterprise. Any scientist wants their work to be viewed and valued elsewhere, not just in their town or country. And that may work in our favor. And perhaps make clear that this has to be a global undertaking somehow.
I'm just going to parse two questions from the Zoom for your response, and then we'll celebrate and conclude. So Ellen poses the question, picking up on what was just discussed, is this type of soft enforcement and power likely to be sufficient? It's like we're doing it through guidelines and things that aren't straight up law with criminal penalty. What about private industry? What about things that are outside the academy? That's one question from Ellen. And then Michael poses a question, and I'll condense it. Do you think as we understand more about how infectious agents work, as we gain more knowledge, does that make it easier to pose clear red lines? Meaning, is the whole red line approach advantaged by increase in knowledge or not? So curious, those are the two questions from the Zoom.
So thanks, Ellen, wherever you are. I think the quick answer to your first question is no. These soft power measures are certainly inadequate alone. Will they be useful in simply reducing the likelihoods, the numbers, the odds? Possibly. That would be my hope. It's maybe an aspiration in some people's mind, but that would be my purpose, is in simply reducing likelihoods. And the second issue was
as we learn more about infectious agents, does it help us make better red lines or the other way?
I think the answer to that one is yes, because it helps in enhancing our ability to predict the possible outcome of a proposed experiment. Part of the flaw in a red line or a standard or an aspiration is, and I thought somebody was going to mention this, is that oftentimes we don't know what's going to happen with an experiment. We didn't intend to do this, but nonetheless, look what happened. And that will become possibly less common as our ability to predict on the basis of, primary information, data, and understanding of the system improves.
Great. Thank you very much. Please, everyone, join me in thanking David for a wonderful talk and discussion.